start right away with our today's lecture. Today's lecture will be about the activation of T lymphocyte, and I will spend some time uh, for you guys just to refresh your memories with me related to molecular immunology, and also we will pin down. We will going to see the mechanism of activation of lymphocytes, particularly the T cells and the beta cells, in more advanced details. And we will try to see what other invisible factors are involved in the activation of the lymphocytes. Okay, so let me take you to the first slide. So generally, we know, just for your, just to recall our memories. Generally, we know that a lymphocyte or a T cell, for example, is activated with the help of an antigen presenting cell. The antigen presenting cells, with the help of the MSC complex, आपने पढ़े, there are two types of MSC complexes generally, MSC1, MSC2, वैसे MSC3 भी होता है. The talk between the TCR and the MSC allows the transfer of information from the antigen presenting cells to the T cells. And then what will happen? You know, the T cells will start proliferation, they start differentiation, colonal expansion. Not every T cell is supposed to divide. The one T cell which get the information, it should divide. This is, you can consider or call as colonal expansion. Increasing the number of just one particular T cell. And then of course, the differentiation. The differentiation helps the T cells to become different, to acquire new functions, and to start doing a newer and different type of jobs. And of course, at the end, we should have the memory cells. So these are the factors which we already know. The T cells recognize and get activated out of the APCs. And it no longer stays as a naive T cells. And of course, there are the partners, other cells, including the beta cells are there as well. So let's go further. I'm sure this is a good enough recall, because you might fed up of listening the T cells and the beta cells, the T cells and the beta cells again and again. But I'm sorry, I mean, we have to proceed further with these things. Okay, so let's try to be easy on these difficult things. So there are two signals which are required, necessary, important for the activation of T cells. Generally, earlier we just thought there should be MSC, you know, MSC, it should be present on where? The MSC should be present on the APC. So this cell is supposed to be the APC. And this cell, this one is the T cell. So earlier we know that the interaction between these two cells is executed between these two protein receptors. The one is called MSC. If it is an MSC class 2, you can guess it's an MSC class 2. It binds with the TCR, the T cell receptor, and you can see this brown, this little brown stuff here. This is the part of the trouble. This belongs to the bad thing. This is the antigen. And that antigen is presented on the MSC and then sensed and captured by the TCR. But this combination is not good enough. Remember, there is no very strong force of attraction between these two protein molecules. They are not very much tightly connected with each other. They need some additional support from some additional protein-protein interactions. It is required, it is important. So the one signal which gives the go-ahead to the TCR, to the T cell, comes from the interaction between MSC and TCR. So MSC TCR के interaction से the first signal means that the first go-ahead signal comes from there. The second go-ahead signal comes from the other protein-protein interaction. And the most famous of them is the interaction between the B7, it's a membranous protein present on the surface of APC, and CD28. It's another type of protein present on the surface of the T cell. This type of interaction helps to establish the earlier connection stronger. These two are not very much tightly linked with each other. There are loose interaction between these two protein-protein molecules. The additional interactions help these two cells to stay together so the MHC can efficiently pass on the information to the T cell. This is called co-stimulation. This is the second signal. There's a first signal here. This is the first signal, and this one is the second signal. If these two signals are there, then we can say that the T cell will receive a complete instructional go-ahead to proceed further for the process to make effector cells. It will increase its number, proliferation, colonal expansion, differentiation. It will create different type of cells, and including the memory cells. So that is how it starts from here. There are different types of proteins and their counterparts exist on these two types of cells. So as a good molecular immunologist, you should know at least some of them very well. The first, you should know that the ligand or the protein which you are talking about is present on where. You know, this is the ligand, very famous, class 2 MSC, you know, class 1 MSC. These are called as the ligands. They are present on where? They are present on antigen presenting cells. We know that. They are responsible to connect themselves with the T cells with the help of TCR. But they need some additional signal. They need some additional support. Co-stimulation from other proteins. Okay, don't forget this thing. Now, this is called as the ligand. This is technically an important stuff. This is called a ligand or a ligand. Remember, in cell signaling, a molecule, an external molecule, is supposed to activate this cell. For example, this thing is a chemical compound or estrogen, for example. It can stimulate the growth of this cell. This cell can receive this stimulus. The cell can receive this stimulus with the help of receptors present on the cell surface. So this thing is called as the receptor, the one which traditionally receives the information. This thing which will going to act as a message, which will going to act as a signal, is called as the stimuli, or more famously it is called as the ligand or the ligand. Whatever the pronunciation is correct, you can use it. So the ligand is something which is sending the signal. It binds with another protein, and that protein which receives that information is called as the receptor. And then you can know, you should know that it will trigger a signaling cascade. It will trigger another molecule, that will trigger another molecule, that will trigger another molecule, and it will end up in some sort of function. In some sort of function at the end of the day. That is how signaling cascades work. Now, just recall the last slide. The information is coming from where? The information is coming from antigen presenting cells. So, and that information is sent or communicated with the help of MHC. So the MHC is the ligand, because it is going to share that information with the next member. And TCR is the receptor, receiving, the one who is receiving the information. That is how traditionally we consider that which one is the ligand and which one is going to receive that information. There are certain associated co-stimulating protein molecules are out there. You should know some of them very well. CD8, you can guess. CD4, you know, with T4, CD8 cells. CD28, You should know. You should know CTLA4. You should know PD1. At least you should remember these few important protein receptor molecules present on the T cells. They are involved one way or another in certain important signaling in cascades. But PD1 is supposed to interact with PDL2. That is very important. PD1 is supposed to interact with the PDL1 or PDL2. CTLA4, please remember this one. CTLA4, which is present on the TCR, or which is present on the T cell, is supposed to interact with the B7. CD28 can interact with the B7. So B7 can interact with the CD28, and it can also interact at the same time with the CTLA4 as well. This is very important information for you, which might make you a better undergrad student. One B7 present on the antigen presenting cell is able to communicate with CD28 as well as with CTLA4. Just remember this thing for some time. I will use that information in the upcoming slide to unlock another concept. So that is how they're able to produce all these cells. You know, naive T cells, then become effector T cells, memory cells, and regulatory T cell. You know, the regulatory ones, the ones which are supposed to delete the extra cells produced as a result of immune reaction. They are required, they are important. There are other cellular proteins also involved in establishing the connection between a dendritic cell or an antigen presenting cell and T cells, in addition to the MSC. They are called as ICAM 1, ICAM 2, and that is good enough. You just need to remember that in addition to MHC TCR interaction, in addition to B7 CD28 interaction, we need integrins. The cellular protein, the cytoskeleton proteins, they are very, very important signaling proteins because it helps the cells to sense what is present outside the cell. So these proteins are also involved in stabilizing the connection between the T cell and the antigen presenting cells. So what will happen if the stimulation is not there, co-stimulation is not there? For example, this is a resting situation when the co-stimulation is not there. You can see there is an MSC molecule here. You can see there is a TCR is here. It is communicating with each other without the support. There should be, what should be here? There should be B7 here. There should be B7 here. It should communicate with CD28. It's not here. So that will not going to trigger the process of proliferation and differentiation in the T cells. The T cells, if it is naive T cell, it will stay as a naive T cell, means no response. So that might be a reason of disease in certain patients, that they are not carrying enough co-stimulators in their antigen presenting cells. In this particular situation, you can see after establishing a connection between MSC and TCR, you will get an additional stimulation between B7 and CD28. If the B7 CD28 meets with each other, it's a second signal that will complete the connection. It will make them, complete. वो जो कहते हैं ना दोस्तों से, you have completed my life, something like that. So that TCR can speak with the same emotions, that you have completed me. It triggers multiple things, including the secretion of certain important cytokines. Please remember, it is the cytokine called interleukin 12. It's a small peptide, small molecule, like small protein. It releases interleukin 12. The secretion of interleukin 12 actually triggered or push the cascade. It changes the morphology of the T cells, and eventually it will lead to the effector cells. So a proper combination in the presence of co-stimulators completes the connection, triggers the secretion of cytokines, interleukin 12, and which will push with the kama that those T cells down the hill. And after that, it is almost committed. It will start proceeding and start proliferating and producing the effector cells. So do not get scared of this picture. I don't want you to memorize this picture at all. I just want you to see that one cell receives information from multiple receptors through different types of receptor, from here, from here, from here. All these receptors are generally present on the cell membrane. For example, you can see RTK, receptor tyrosine kinase, GPCR, it's called G protein couple receptor, something. So there are different types of receptors present on the cell membrane. Don't worry if you can't memorize all these names. But here you can see another receptor here. Oh, cytokine receptor. We should be interested in this one, Hannah, because in our course or in molecular immunology, we heard a lot that a cell is releasing the cytokines, which changes the destination of the target molecule. How it is possible? The target molecule should be the target cell should be able to catch the cytokines. So the cytokines can bind themselves with the help of cytokine receptors. And after that, they trigger a signaling cascade, which we might see, not now, but after that. There are other signaling cascades out there. One of them is this one, PI3K AKT signaling pathway. You can see PI3K, and then it was written AKT, and then it was written AKT. So it's called PI3K AKT signaling pathway. The reason why I am actually highlighting this pathway is called a survival signaling pathway. Once this pathway is activated, you can trace ये कहाँ जा रहा है. You can trace where it is going. It is going to here, PKC, then NFK beta, and then into the cells. It will activate the genes, and those genes will help the cells to sense that I need to survive. What that means? The cells will feel everything is fine. The cell will feel I have good enough food. The cell will feel I need to flourish. I need to divide. I need to proceed with the cell division. So the overall, the sense of the cell will be like that. I have everything if this cascade is activated. And this cascade is activated in case of abundant supply of food and if everything is fine. And that triggers the activation of certain genes, and those genes will help the cells to grow, to proliferate, to differentiate, and to produce more type of cells. That state of mind is important for the cells if the cell wants to proceed for the cell division and for the differentiation. Now, there's a thoda sa reh gaya, hosla. Now think about, I'm just going back, okay? वैसे मैं going back क्यों कर रहा हूँ? Going forward में भी आ जाएगा. Now this type of thing you should know. Now imagine the state of naive cell, the signaling state of naive cell after the connection, the complete connection with the APC in the presence of co-stimulation, it triggers the very important survival signaling cascade within the naive T cell. And that naive T cell, जिसको पहले लगता था कि मुझे कुछ करने की जरूरत नहीं है, I'm fine, I don't need to grow, I'm okay, something like that. Suddenly, it start feeling that I need to proceed for the cell division. I need to proceed for the cell division and differentiation. I should not give you this example, but it is more or less like a motivational speaker, you know? You just listen a 5 minutes talk of somebody and then you start working. ऐसा होता कोई नहीं है. I don't know. मुझे थोड़े से weird लगते हैं ये. So कि ये कैसे possible है कि आप किसी को बोलेंगे और वो उसके बाद जो है उसकी जिंदगी बदल जाएगी. Anyways, कुछ करते होंगे ऐसे. लेकिन सबसे अच्छे मोटिवेशनल स्पीकर जो है ना वो फैमिली में होते हैं. You know, they are present in our families. वो जब आपकी बेइज्जती करते हैं ना, सबसे अच्छी मोटिवेशन तब मिलती है to become something. So now let me share this thing. So you can see once the complete connection is established, I'm sure I will be very happy if you're able to decipher this picture comfortably. Look at that. This is the D cell, dendritic cell, an antigen presenting cell. This is the T cell, MSC, TCR, combination, co-stimulation between B7 and CD8. All things are set. This is integrins, ICANS, these one. All things are set. What will happen? It will trigger PI3 AKT signaling cascade. It also written RAS MAP kinase. It's also a survival pathway. So once this one is done, that will going to increase the production of BCL and BCL2. That will going to increase the secretion of interleukin 2 and expression of interleukin as well as the 2R. It will going to increase the cyclins and decrease the cell cycle inhibitors. And it also affects some other mechanisms as well. Just go through some of them. It increases the production of BCL, BCLX and BCL2. These things help the cells to not to think about the suicide. Okay? ये सेल को बताता है कि मुझे जिंदा रहना है. Means I don't need to go for apoptosis. So apoptosis is not in a thought process at all. Survival. It's time to live. The secretion of interleukins that helps in the cell divisions and cell proliferation. Look at that. Cyclins are increased. Cyclins help the cells to pass on the cell cycle. Cell cycle checkpoints se aage jana and proceed with the cell cycle and go further for the cell division. Decreases the cell cycle inhibitors. Har wo cheez, everything which is stopping the cells to proceed for the cell division is actually blocked. So overall, imagine the state of the cell. It's just like in the, just like what? Just like what? It is just like a state of the person in the first love. I have energy. I can conquer the rest of the world. I will go, I will proliferate, I will differentiate, and I will go and create all sorts of wonderful things which is required to execute the function. So that is the state of the cell, of the lymphocyte at that time.